Hello and welcome everybody. Today we are going to solve the May-June 2025 Paper 2 Variant 3 Paper 2 Multiple Choice. Let us start with the first question in writing here. It's telling you here that the diagram shows an aquatic plant in light, and as you can see that plant is surrounded by light and it is giving out bubbles containing oxygen gas. It's telling you here which characteristics of all living or things it is demonstrated in this diagram. So in this diagram, what is really demonstrated, as you can see here, is letter A, breathing, because all living organisms breathe. We are as humans breathe, but we cannot carry out nutrition. Although if the all phrase was not included, nutrition and B and C, nutrition and photosynthesis, would be correct. But in this case, it's not correct because it is not nutrition and photosynthesis are not shared by human beings and by other creatures. Okay, like for example animals, they don't have photosynthesis. Now question 2, diagram shows 4 organisms. What is a prokaryote? As you can see here, I had illustrated for you a diagram. Prokaryotes are bacteria, and bacteria have flagellum, and remember that the flagellum that the bacteria have is also in another organisms, but like that organism in letter C. But in order for us to identify that either A or C is a bacteria, we will look into the inner plasmid part. And as we can see here, we can find a circular DNA sitting inside the plasmid, which depicts and shows that A is a bacteria, of course. So C is wrong and A is the correct answer. In question number 3, it's telling you what happens when plant cells are placed in a sugar solution that has a lower water potential than the cytoplasm of the cells. Okay, so basically you're going to place a potato cell, okay, or plant cell in a beaker, and that beaker is placed in and containing a sugar solution, and it has a lower water potential, meaning that there are more sugar molecules present in the solution compared to the water molecules. So meaning that the water will move out of the plant cells down the water potential gradient by osmosis into the beaker containing the water. So basically water will move out of that plant cells, it will move out. So this means that the cell will lose water and it will become flaccid. Now coming to question number 4. In a DNA molecule, bonds between pairs of bases hold 2 strands together. High temperatures can break these bonds to form 2 single strands of DNA. The base pair C and G is held together more strongly than the base A and T. So basically the question wants to deliver for you an idea that cytosine when paired with guanine are linked by three bonds, and adenine and thymine are linked by double bonds. So basically and by common sense, cytosine and guanine have 3 bonds, so they will need a higher temperature to break down these 3 bonds compared to breaking the 2 bonds, because the double bonds can be broken down by a lower temperature compared to the one that is linking cytosine and guanine. So the answer here for question 4 is letter C, DNA molecule 2, because there are more A bases present. And for us to understand that part, you can just look into the diagram I had illustrated for you. As you can see here, DNA 2 was chosen because it has more adenine to thymine bases compared to DNA 1, which has cytosine to guanine bases. Okay, so you need to keep that in mind, and please don't forget such an important concept. It's telling you here which arrow identify the substrate and the cause of process are. So basically we can understand that in here the substrate is really identical. Like see that like crocodile face is the same as that, so that can be fit here easily and without any problems. Now coming on to the next question, either it's caused by an alkaline or acidic condition. Well, if it's acidic condition, it will denature the enzyme and the enzyme won't work again. But in here, for us to understand it is alkaline, it was only changing the shape of the active site. It did not denature the enzyme completely, although it only changed the shape, but that enzyme can work in the future because it has not been damaged by an acid, so it's still not denatured yet. Okay, now moving on to the next question, number 6. What is the balanced chemical equation for photosynthesis? Now, the answer, of course, is C. It's something you need to memorize. It's nothing like a hard. Okay. Question number 7. A potato was removed from a potato plant. A 5-gram cube of potato was cut and put into a beaker of distilled water. What is the most likely mass of the cube of the potato after 2 hours? So a potato plant placed in distilled water, meaning that the potato plant is placed in a higher water potential solution, meaning that the water will move, the water outside the potato tissue will move into the potato tissue, and it will cause the potato to increase in mass, expand and increase in length. So basically, of course, mass will increase, so the answer will be D. Question 8. Diagram shows direction of net movement of two substances across a human placenta. It's telling you here which tissue represents maternal blood and which substance is identified correctly. For us to understand this question, basically you need to grasp the idea that now your mother or the mother will deliver for the fetus, what will deliver for the fetus nutrients, like for example sodium ions, like for example glucose, like for example anything that can benefit the baby to survive. And the fetus in return will give the mother what? Will give the mother the carbon dioxide, the waste products of metabolism of the fetus in his mother womb. Okay, so now tissue that represent the maternal blood, of course it's tissue B because it is providing. Nothing else can provide a baby unless his mother. So a fetus or a baby is provided nutrition by his mother. So basically tissue B, it will be the correct answer for maternal blood. Substance is identified correctly, of course B in here, substance 1. Okay, what will happen? It is giving, what is giving urea, and we need to understand that the mother here is giving urea. Okay, so that was all about that question, question 8. If you have any questions, leave it in the comments section. It's saying here which structure parasite mesophyll cell. It's something mainly memorized, and it's letter B, not very hard. It's telling you here bleeding gums, slow wound healing, which is basically scurvy. So the lack of nutrient is vitamin C, so your answer here is D. With digested food molecules move into the cells of the body. Okay, move into what? Into the cells of the body, where they are used and become part of the cells. What is the definition of this process? It's called assimilation. And for us, like really to understand this part, we can just remember with me, can you remember with me that in variant 1, the same question came. And we told that assimilation, it is the uptake, okay, uptake and use of nutrients by the cells. Uptake and use of nutrients by whom? By the cells. So just keep this in mind, because this is really a repeated idea, and I see that Cambridge really focuses on that part. Question number 12 is telling you here the diagram shows part of the human digestive system. Okay, in which part of the protein digestion begins? Protein. So basically protein will be broken down in two parts, and we'll give you here a drawing for you to understand. Now for us, now the protein, it is broken down by two enzymes, pepsin and trypsin. It's broken down by what? Pepsin and trypsin. So basically the pepsin works in the stomach, trypsin works in the duodenum. So is the stomach BH2, the duodenum BH8, which shows that BH2 is acidic and BH8 is alkaline. Okay, so this is just a reference for you to understand. So basically protein digestion, of course, will start in the stomach, and that will be broken down by pepsin, and pepsin will be involved in such a case. Okay, so now let us move to the next part. Question 13. Diagram show cross section of a leaf, a root and a stem, and it's telling you which one shows the xylem. So basically I told you before that the xylem here, it's a it's non-living tissue. This is an information, but in this question, in order to solve it, you need to memorize the drawing. And if you in case you didn't memorize it, no problem, you can still figure it out by using my trick. Now xylem, of course, it will be the bigger part in the vascular bundle, as you can see here in the roots. Okay, in the roots, can you see that star symbol marked in yellow highlighter? That star symbol, it's larger. So we can say that this is a xylem because it delivers more water at a higher pressure. Now here, when you're comparing 5 and 6, who is larger? Of course 6 will be larger than 5. If we here like see it closely, the 6 will be larger than what? Larger than 5. So of course 6 is what? 6, it is the xylem of the stem. Number 14. Which structures in human and plants normally carry amino acid? Of course, arteries and phloem. You tell me why phloem? Because xylem only transport water and mineral ions, and it is a common mistake where students like miss think this part. They think that the xylem transport amino acid, which in case it's not correct, because the xylem only transport water and mineral ions. Also in here, why arteries? Because arteries are the ones which deliver the blood to the stomach, to the muscle fibers, tissue, and so on. Question 15. Through which pathway does blood leave the heart? He's telling you here through the arteries, from the ventricles, okay, or through the arteries from the atria, or through the veins from the atria, or through the vein from the ventricles. As you can see here, for this question the answer is B. So how through the arteries from the ventricles? Well, the blood will move from your left ventricle, which is marked as 2 here, from your left ventricle, and the blood will move what? And the blood will move, right? Will move from 2 flowing through the aorta, which is the largest artery in the human circulatory double system, okay, and will move out of the aorta at high pressure to all body parts. Number 16. It's telling you what is the name and the function of the cell labeled X. Labeled X basically is a phagocyte, and here is a picture of a lymphocyte for you just to memorize and to study it. And actually it's a memorized concept, just you need to like figure it out. Of course phagocyte does not produce antibodies because phagocytes carry out phagocytosis and it engulfs pathogens. And we can remember this by a trick I told you before in June variant 1. We can say that phagocyte means what? Means pathogens. And where is the similar part? Well, the similar part is right here. The similar part is in right here. Okay, so just memorize these for you to understand and for you to study it. Okay, now moving on to the next question. Question 17. It's telling you here which row shows the effect of cholera on a human body. So the movement of chloride ions here, it will be into the small intestine, okay. An effect of build up of chloride ions on water potential, it will decrease. An effect on volume of water leaving the body through the anus increases. Why this one increases? This is a many like questions a student ask and get confused about it. Why that really decreases? Because as you can see here, this is my blood canal, okay. This is my arteries, okay. This is my arteries, and the water will flow from the arteries into the gut where dehydration occurs. So less water potential inside the arteries, so less water molecules will be present in the blood, and water will move out of the blood into the gut. This causes dehydration. Question 18. Don't just be like scared by the graph, it's so easy, nothing like to worry about. Structure attached to alveoli, okay, it will be here at 2. Why at 2? Distance from the larynx is greater and relative diameter is lower. So distance from the larynx, of course it's greater, because as you can see the larynx is some part in here, but the alveoli is some part in here attached to what? Attached to your bronchioles. Attached to what? Attached to your bronchioles. The alveoli is here marked in yellow. Now a structure containing cartilage, of course the distance will be much like nearer to the larynx, because as you can see now, can you remember and recall that cartilage, they are rings which are forming rings like that, rings. Why? Preventing what? Preventing the airways from collapsing and to ensure that they are strengthened. So basically it's like a very very simple easy question and nothing to worry about in such a question. Very easy. Okay, question number 19 is telling you here which process in plants uses energy from respiration. Respiration meaning cell division. Why cell division? Because basically osmosis is a passive transport. Photosynthesis does not use energy from the respiration. In fact, photosynthesis releases energy in plants because it uses carbon dioxide and water producing glucose and oxygen. And transpiration is just evaporation of water, and evaporation of water does not require any energy from respiration. It only requires heat. Question 20. Which sequences of statements explain the build-up and removal of an oxygen debt? Basically, of course, I'm gonna cancel C and D, because if you look into 3, how gonna glucose be converted to lactic acid without even an aerobic respiration occurring? It is impossible, so C and D are out. Now A and B, for us to understand the answer first is A, okay? Now what we can say, now A and B here, they share this common confusion part, but all other numbers are same. Who is first? Who is gonna come first? 2 or 4? Of course, the answer will be 4, because lactic acid need to be building up in the muscle. For example, suppose this is a muscle, okay? Muscle. This is a muscle cell. Lactic acid need to be built up in the muscle, okay? Before what? Before breathing occurs. Because like just suppose if there's no here lactic acid, okay? Like just suppose if there is here no lactic acid and oxygen will enter. Tell me it will react with what? Like when oxygen enters, it will react with what? Nothing. It will react with nothing. Basically, it will react with nothing. But if I have my lactic acid, well, so now I gave a gate for the oxygen, I gave the chance for the oxygen to enter, so that the oxygen can react with that lactic acid and will be broken down easily and simply. Question number 21. What is filtered out of the blood in the glomerulus and into the nephron? Glucose, yes, and urea, yes, because glucose and urea, they are small molecules, okay, and they are not large molecules such as proteins, so of course both, yes, both will pass and there will be no any problem. What can be broken down to form urea? Of course, to form urea, it is amino acid, and I told you before in variant 1 the amination, okay, of what? Of excess amino acids. The amination of what? Of excess what? Of excess amino acids in where? In the liver. Of excess amino acids in what? In the liver. Okay, moving on to the next question. This is my lovely lesson, nervous system. It's very easy. Diagram show a simple reflex arc. As you can see here, what is the name of the structure labeled B, Q, and R? So B, of course, it's sensory neuron because it's linked to your sense organ, which is the finger. And you tell me what it senses or what it detects. Well, it can detect pain, okay? It can detect pain. It can detect pressure, okay? It can detect like high temperature, okay, or touch. It can detect touch. It can detect many things, okay? Just in case you need to just know them in case they came as a multiple choice question or like paper 4 question. Question 4, of course, it will be the relay neuron because it connects your sensory neuron to your motor neuron, which is letter I, I mean. Okay, so R here will be the motor neuron, and the motor neuron basically it is attached to the effector, and the effector will tell me what is an effector. Well, an effector, it is a muscle, and that muscle, when that nerve impulse will be transmitted into it via the motor neuron, it will cause an effect. The muscles will contract immediately to remove away from the source of danger, which can be a fire, a stink by an insect, or even someone if touched you, okay? Now moving on to the next question, 24. The diagram shows a cross section of an eye, and here in 24 is telling you which row is correct when a person views a near object. So here, this is the conditions for far object. Suspensory ligament will tighten and ciliary muscles will relax. So in order for us to like answer when we are viewing a near object, it will be the opposite. So suspensory ligaments in here will what? As you can see here that the G of the suspensory ligament, so they will be pulled less. They will not be tightened anymore. They will be pulled less. They will be slackened, okay? And the ciliary muscles will contract. Ciliary muscles, which is letter E, that will contract. As you can see here, contracts. As you can see here, it contracts. And for G, it is pulled less. And F becomes thicker, of course, because, like for example, if I am having here a circle and I'm pulling it by 2 stripes like this, of course, and this is an elastic ball, for example. So when I'm stretching it, well, after stretching, it will be like this. After stretching, it will be like this. But if I'm not stretching, it will be like this. So the one that I'm referring to in the red color, in this color, okay, this is the case of the question. This is what he wants you to say and what he wants you to imagine in the question. Okay, so of course refraction of light will be more because more light can enter now into your eyes, and this is a common concept like in physics. If you took physics, the subject physics in IGCSE, you will understand this more. Okay, but like it is like this one reflection does not have to do anything with physics and biology. Just memorize it and you will be okay. Now, which hormones increases the blood glucose concentration in humans? Of course, adrenaline and glucagon. You will tell me how glucagon, because basically, as you know, that glucagon, what glucagon breaks down glycogen, and breaking down glycogen. Okay, it breaks down what? It breaks down glycogen, right? It breaks down glycogen. So when glycogen is being broken down, it releases what? It releases for you glucose. Okay, you will tell me now how adrenaline does so, because adrenaline, it is a hormone released from adrenal glands. It causes more energy to be released by respiration, so of course glucose need to increase as well. 26, a question is telling you here. Okay, a question is telling you a shoot tip was placed on a agar block. Substances can diffuse through the agar. Light was shown on the shoot tip from one direction. Here is your shoot tip, here is your agar block. After 3 days the agar was cut into half to form block X and Y. Block X and Y were placed on the stem of shoots that had the tips cut off. The agar blocks were kept on the shoot stem for 1 week. And as you can see that the light, see the light here? Can you see the light or no? The light is pointing to that direction, to the left direction. Okay, so basically here the answer will be C. You will tell me, but why the answer here is not A, or it's not like B, or it's not like D? First of all, it's not D because it is impossible for a plant to stretch against the direction of light. Like if it like moving to that direction, it needs to bend to that direction. If the light is moving to that direction, so the plant need to move to that direction. So like basically by common sense, D is like invalid at all. D is invalid at all. So the most common and suitable answer for this question is C. The reason is C because as you can see here in X, the light maybe had diffracted or diverged. Okay, he's saying light was shown on the shoot tip from one direction. Now he's telling you that the shoot tip, okay, after 3 days, the agar was cut into to form block X and Y. Block X and Y were placed on the stems of shoot that had the tips cut off. So now the tip cut off, meaning that Y will not move in either direction because it does not have a shoot tip. There is no oxygen that can control the shoot tip to go like in the left direction or in the right direction. There is nothing. So like C and B until now they are true. The reason why C is correct because X, okay, here X it's still on the remain side. Like see here X, here it is there, right? And while that X is on the other side, that part it is in the opposite direction, so we needed to keep it bending it to the right, okay, against what? Against the light source. Okay, so this is like just a tricky question, you need to understand it well. 27 is telling you here diagram shows reproduction in potato plant. Potato X was planted into the ground and a plant grew from it. The plant then produced the potato Y. Which statement is correct? Y was produced by asexual reproduction, of course. X and Y are genetically different, of course. That is incorrect because basically they had carried out asexual reproduction. Y was produced by asexual reproduction, no, because there was no like cross-pollination, self-pollination, there was nothing like that. Okay, Y was produced by the fusion of gametes. No, there is no sperm, there is no ovum, or there is no like egg cell for them to fuse together. So the basic answer for that question will be B. Which statement about pollination is correct? Which statement about pollination is what is correct? So just telling you here, self-pollination increases ability of a population to respond to changes in the environment by increasing variation. Of course no, because cross-pollination does this part. Self-pollination increases ability of a population to respond to changes in their environment by reducing variation. Of course no, it does not alter variation in that sense. It does not alter variation in that sense, okay? It does not increase it. Yes, it reduces variation, but it does not increase the ability of population to respond. It decreases the ability of the population to respond. So let's just check C. Okay, until now it's okay, but by increasing variation, no, it is incorrect because the one that increases variation, it is cross-pollination. Self-pollination reduces ability of population to respond to changes in the environment by reducing variation. Self-pollination reduces ability of a population to respond to changes in the environment by reducing variation. Yep, this is a true and a correct answer. And the reason why it's correct in that part, because in the sense of it reduces variation, and at the same time it reduces ability of a plant to survive or to respond to the changes in the environment. So basically this question, like, it's really helpful for you to memorize and like just to have a concept of it. Now let me write it for you and make it clear. Now, cross-pollination. Cross-pollination. Cross-pollination, it increases your genetic variety. It increases what? Your genetic variation. And it increases your chance, as a plant for survival. For what? For survival. Okay, so now moving on to the next question. I hope that it makes sense and it's like clear. Okay, now moving on to the next question. Well, the next question here is telling you the graph shows changes in the thickness of what? Of the uterus. In the thickness of what? Of the uterus wall during a menstrual cycle. Now, for us to understand that question, it's really basic. Just memorize the drawing in your book, in your textbook, or in Save My Exams, and it will be okay. So, as saying to you here, when is the last day of menstruation? Well, it will be day 4 in this part. Day 4. Just let me like analyze it for you properly. It will be here. It will be here. Day 4. You will tell me why we chose this part like specifically. As you can see with me here, that the line started first to decrease, right? It started to decrease. It was first here at high. Before it was first here at what? It was first here at high. But after that it starts to what? It starts to decrease. It was here at what? At high. But now it starts to become what? To become lower. Lower in thickness. So that's why the last day of menstruation was day 4, not day 7, not day 8, because day 8, as you can see, the thickness of uterus wall lining was increasing, meaning that the menstruation had ended a while ago. Different version of a gene or alternate version of a gene, it is the same as an allele, because chromosome, of course, chromosome is not a different version of a gene, and chromosome is found in the gene. Length of DNA, it's found in the gene. Protein, of course, no, because protein consists of amino acid that are expressed. Now, the diagram shows the cells of a mammalian embryo shortly after fertilization. What is the correct description of these cells? Stem cells undergoing mitosis. Of course, they will undergo mitosis. They cannot undergo meiosis, because meiosis, you need a female gamete and you need a male gamete for them like to produce a zygote or to occur and allow fertilization to occur. So this one cannot be an answer. So 31 is your correct choice. Now another lovely pedigree question is telling you here diagram shows inheritance in one family of a 6-linked, okay, bear with me, 6-linked recessive disease. So now the chromosome will be carried on what? On the X chromosome. Okay, to be carried on what? On the X chromosome. Now let us analyze. It's telling you here which are heterozygous for this disease, 2 and 3. Well, 2 here and 3. You will tell me, but why 2? I will tell you, because 6-linked disease, it is more commonly found where? It's more commonly found in females, right? So like the female, now for example, if I am having a disease like hemophilia, okay, hemophilia, and hemophilia, in fact, it's like an A2 disease, like it's found in A2 syllabus, and it is 6-linked. Okay, now for example, if I say that I'm giving this hemophilia like a letter A, if that person or that female has a disease, okay, so XA and XA. Okay, amazing. Now you are saying that number 2, it is a carrier. That number 2 female is a carrier female. She is not infected, and the evidence is this diagram. The evidence is this diagram, where as you can see here, the evidence is this diagram. You have XA, okay, she has one allele from the hemophilia, from the disease, but the other allele is the small a, meaning that it's recessive allele, so it is not infected, it is only a carrier. So now it is married with what? With an uninfected male, and it produces an affected male. Okay, you will tell me now for me how that affected male arrives. Okay, basically because the female passed an XA, which is this one part, it passes an XA small, and the male passed the Y chromosome to form a boy. So by chance, in sex-linked disease situations, the female will give the male, okay, the male offspring, it will give him what? It will give him that X. We give him that X, this one. And basically, of course, the man will provide only with the Y, because you are not a female, you're a male, so you will be provided with a Y. So basically now you get an affected male. With this is the reason of it, like if you're asking yourself, if you are wondering how that happened. And the same situation here of 2 is the same situation of 3, because like biologic sense, how that 5 became infected, like how it became infected, not by luck, of course, by this concept. So this concept, it is the whole concept of the question. It is the whole concept of what? Of the question, okay? And that is like sex-linked recessive disease idea. And hemophilia actually is a disease idea, and it's from the A2 syllabus, just for you to understand and to know in your future. Now it's telling you here, which row describes continuous and discontinuous variation? A lovely question. Of course, continuous variation is shared by the combined effect of the genes and the environment, while discontinuous variation is only caused by the effect of the genes and something like just to memorize, nothing about. Okay? Now, question 34. Which statement describes an adaptive feature in an organism? It helps an organism to survive and reproduce in its environment, of course. It always shows continuous variation. No, not always. Actually, it does not show continuous variation. Like for example, a penguin can have like an adaptive feature of diving into deep water to catch fish and catch shrimps to eat and to survive on. Of course, it's not like continuous. Of course, it's not like increasing his depth. He cannot like dive to 1,000 meter underwater. This is not gonna occur. Like it only has specific limits for it to dive into the water. It is inherited, of course it is inherited, because that adaptive feature is caused by evolution. And evolution, it is the change in a feature over many generations by natural selection. So of course it is inherited, of course. Now 35. It's telling you here, the graph shows changes in percentage of bacteria resistant to antibiotic X. What causes a change shown in the graph? Of course, it's natural selection, not meiosis, because meiosis causes variation in humans, not in prokaryotes. Random fertilization also occurs in humans, not or in animals, but not in prokaryotes. Artificial selection involves the male acting as a selective desire. The male selected this, like for example, this cow was high milk yield and this bull was high milk yield, and made them together. But in bacteria, we are not mating one bacteria that is male with another bacteria female. So of course it's natural selection. 36 here, which percentage of energy loss producers and secondary consumers? Here is producers and here is your secondary consumers. Of course, percentage of energy lost is 99%. It's something to memorize, actually. It's not like a fact or anything hard, okay? And just you can calculate it too, okay? Like 1000 minus 10 over the 1000 times 100, and you will find your answer 99. Just cross these like that, like that, like that. 1 minus 10, it will be negative 9 times 100. Negative 9 times 100, it will be for you 99. Okay, so just this will be the answer, and something to memorize, nothing like hard in it. Now it's telling you here, which process in the nitrogen cycle can be caused by the lighting and bacteria? Of course it is nitrogen fixation. By what? Nitrogen fixation. So of course the nitrogen can be caused by lighting and bacteria, and bacteria is nitrifying bacteria. Bacteria is what? Is nitrifying bacteria. If you will tell me why the answer is not like C, because he told you here clearly that there is lighting being involved. Lighting is an important factor for that nitrogen fixation to occur. So 37 is crucial. Now question 38. Which phase in population show when death rates are higher than birth rate? Of course it is letter D, because in here this is the lag phase, this is exponential or log, and this is a stationary, and this is a death phase. And you can just check it out here, as you can see, like in here in the notes, you can see that this face, it is after the stationary face, which is after letter C. So the answer for that question is letter D. Answer for that question is what? Is letter D. 39. What is a disadvantage of a large scale monoculture of crop plants? What is the disadvantage? An increase in risk of disease destroying the entire crop. Why? Because like if there is only a disease, the whole crop will be disrupted or it will be damaged, because remember that cross-pollination is continuous. Cross-pollination occurs at all times. So just we need to keep in mind that 39 will always be the answer D. I cannot say an increase in biodiversity because it would be an advantage, and he's asking here for a disadvantage. A decrease in fossil fuel emissions, of course, this is an advantage. A decrease in use of insecticides, also this is an advantage. So just we need to make sure that we are aware of such a concept. Now, some processes involved in the production of human insulin by genetic modification are listed. It's telling you here what is the sequence of these processes in genetic modification. 3, 1, 4, and 2, right? So 3, human insulin gene DNA is isolated using the restriction enzyme, okay? And of course this one is really crucial because you need to extract the DNA before working on it. You cannot work on the DNA before extracting it. You need to extract it at first. So of course now A and B will be the incorrect options in here. So now it's telling you 3, okay, and it's telling you giving 1, 4, and 2. So 1 basically because insertion of human insulin gene DNA into cut bacterial plasmid. You cannot you cannot tell me that bacteria contain the recombinant plasmid multiply. How? And you didn't even insert the gene into the bacteria. First you need to extract the DNA, extract the gene from the DNA, and then insert that gene into the plasmid for the bacteria, and now that we called it as a recombinant plasmid, and that recombinant plasmid will multiply and will make clones of cells and will multiply over a period of time, and finally your insulin products will be produced easily. So now just let me discuss for you that concept of genetic modification just for you to understand it. I know it's like just something advanced and it's actually from in the A2 syllabus, so if you understood it now, you saved your life and you saved a lot of time in your A2 syllabus. So it's telling you here genetic modification, how it occurs. Now if I have here a DNA, okay, now if I have what? If I have a DNA, okay, DNA like that, a DNA like that, and I extracted a gene out of the DNA, so that gene, okay, that gene will be placed in what? That gene will be placed into the bacterial plasmid. It will be placed into what? Into the bacterial what? Into the bacterial plasmid. So this one is now called what? Is now called a recombinant what? It is called now a recombinant plasmid. So that one is called what? Is called a recombinant what? A recombinant plasmid. Just let me write it here for you so that you can just study it. Recombinant. No, just do it. This is so big. The recombinant what? Recombinant plasmid. Recombinant what? Plasmid. So basically that recombinant plasmid will multiply and form clones of cells, okay, and then lastly it will make your insulin gene. And that was all about that exam. I hope you had benefited from such an explanation, and I hope that you had an idea of how the difficult questions in this exam and the tricks are solved, and everything is really fine. So in this exam only 2 questions are hard, which is the question for this one question for that enzyme. It's very tricky, and another question that was also tricky in such an exam was the question of the 26, and also another question, if you didn't study well, it will be 32. 32 will also be another tricky question, just you need to memorize and keep in mind. And yeah, that was all about solving the paper 2 multiple choice. If you had benefited, please subscribe, like, and share it to your friends so that everyone can benefit from my explanation about this exam, which is the May-June 2025 paper 2 variant 3, and it was explained by the channel CAIE Sciences Mastered. And thank you for your time and have a nice day.